cover the chemical basis of life, chapter 2. So we're going to introduce to biochemistry for life science, why we need to study this chemistry in this anatomy class. Before we understand how the body works, we need to explore the basic building block of matter, atoms, molecules, and chemical reactions, because all our body structures and function depends on chemistry at the cellular level. So for that purpose, we're going to see some definitions, mass, matter, weight, some other definitions is coming. Make sure you write it down and know those things. What is matter? Matter is anything that occupies space. For example, you are sitting on a chair, so you occupy space. Your computer occupies space. So those all are matters. So when you're going back to mass, this mass is the amount of matter in an object. That is a mass. Then weight is the one force of gravity acting on what? Mass. That is weight. Weight will change based on which planet you live. So the gravitational force is 9.8. So that will, depends on which planet. The gravitational forces act upon the mass, provide the weight. Element and atoms. So elements are the simplest type of matter. If you breaking down the matter, what is the simplest form? So it does have unique properties. Atoms is the smallest particle of an element. Example elements are carbon, hydrogen, nitrogen, oxygen, etc. So elements in living organisms, four main elements we're going to see throughout: oxygen, carbon, hydrogen, nitrogen. This make up that over the 96% of human body. So that is including water. Water is what? H2O, 2 hydrogen and 1 oxygen. Then trace elements is not need that much. They are the calcium, phosphorus, potassium, magnesium, etc. Now we're going to see that atoms and subatomic particles. So what is an atom? We talk about atoms are the smallest particles of elements. So this one atom, if you look at that hydrogen atom, it's only one proton and one neutron, one electron. Neutron can be changes based on the isotopes. We're going to talk about that later. See here, in the nucleus should have a proton and a neutron. In the electron cloud, where you see that electron stays in that electron clouds. Neutron doesn't have any charge. Proton has the positive charge. Electron has the negative charge. The next thing we're going to see that atomic number and atomic mass. Atomic number for an atom or element, we talk about this is the number of protons, only looking at the proton, number of protons. But atomic mass is a proton and a neutron. So, but electron, we ask him why we don't count electron. Electron contribute what? Negligible mass. It doesn't count for that. So here in this case, hydrogen has only one proton and one electron. So here carbon 6 proton and 6 neutron, 6 electrons. Electron is stays in that electron cloud area. So here oxygen 8 proton, 8 neutron and 8 electrons. So this how they arrange that. For example, here hydrogen has what, one proton which stays inside the nucleus and one electron which is outside is arranged here. So the first orbit should have two what, to stabilize that atom. So here is only one right now. So one other one here, which is a carbon. Carbon has a first orbit has 2 electrons. Then second 1, 2, 3, 4. So the 4 electrons are there. In order to stable this carbon, you need 4 more electrons. It's not stable right now. So I need 4 plus 4 makes 8. 8 going to be stable. So here another one, oxygen. Oxygen has 2 in that first shell. Second one has what, 6. So 6, you need 2 more to make it 8. So this is one of the other thing about the oxygen is very electronegative. That means it can attract electron towards that, closer to that easily. So you can see that water H2O. So how the water forming, this oxygen, you notice that they need 2 more electron. So the hydrogen need what, 1 more electron. So the hydrogen goes here. So with that outermost shell, there's with 1 electron, they forming that bond between that. Then other hydrogen over here, bonding the form between that. One other thing also, it will go closer to that oxygen. This electron will, like I mentioned that earlier, oxygen has a more electronegative the nature. So it will pull this electron towards that oxygen from the hydrogen. So this oxygen get a partial negative charge and hydrogen get a partial positive charge. Same thing over here. This oxygen pulling that electron towards that from hydrogen to oxygen. So the oxygen get a partial negative charge. So that is the way it's forming that water molecule. Then we're going to see that molecule versus compound. So molecule is 2 or more atoms bonded. Example, oxygen. Oxygen is, it won't stay like this, or always you see atmospheric oxygen like this. Compound is a molecule with different elements. Water is an example, we just seen that. Then we're going to see isotopes. I talk about that isotopes. Hydrogen, beginning I mentioned there is a 1 neutron, 1 electron, 1 proton, but it can vary in that isotopes forms. So proton can be changes, number of neutron can be changes. So here the isotopes have what, 2 or more forms of the same element with the number of protons and number of electrons, but different number of neutrons. So number of neutrons here is 1, here is 2, here is 3. So the name going to be changes based on that, but it's all of them are form of hydrogen. So what is electrolyte, anions and cations? So I'm going to start with the cations. They are positive ions. How they get positive charge? Because they lost electrons. You've seen that Na plus. Na plus is sodium, which is Na sodium. Sodium that lost 1 of electrons, then you get that positive charge. So then other one anions, chloride ion. So this will gain 1 of the electron from here, so it make that negative charge. So if it's become that stable NaCl, which is table salt, when you put the salt in that water, dissociated in the water, you become Na plus and Cl minus. So electrolyte essential for the nerve, muscles and functions. Electrolytes are one: sodium, potassium, these kind of things. That's very important. Then ions, what is that? Ions are atom that has gained or lost electron. When they gain or lose electron, they are going to get what? Charge. Type of chemical bonds. So when you forming that, for example, hydrogen, water. So the water, oxygen has to bond with the hydrogen. So there's a chemical bond between the oxygen to hydrogen. There can be different types. Depends on the electronegativity. I mentioned that oxygen has a more electronegativity compared to hydrogen. So this electron from here attracted to oxygen. So like I mentioned, this has a more negative charge is going to be. So this electron from here pull it towards oxygen. It's closer to oxygen. So because this oxygen is more electronegative than hydrogen. So the closer an atom is to achieving an octet, the higher it is electronegative. So when you look at that periodic table, you see that last one, that's all stable, they are octet. Then the closer to that one is going to be more electronegativity. It can have a characteristics that electronegative more closer, because when you come closer to that, there may be one more electron to what? Become octet. So it is always that looking for that electron. Then next one is ionic, covalent and hydrogen bond. There are different types of the chemical bonds. Ionic is a transfer of electrons. They just transfer. They give that electrons. So like we said, sodium, chloride, this one. Covalent bond, they don't transfer the electrons to one to another. They're sharing electron. They can be polar or non-polar. They just share electrons, but they don't give fully to that other atom or atom. So the hydrogen bond is a weak attraction between molecules. So relative electronegativity of two atoms in a bond, electron transfer, ionic, electron transfer, polar covalent, unequal electron sharing. Some of we study that oxygen pull that electron towards that oxygen from hydrogen. Equal electron sharing, that's a non-polar. There is nothing pulling towards that one, just equally they share the electrons. So that atoms, like I said, the nucleus, they have a protons and a neutron. Proton has a positive charge, that's a nucleus. Outer there is a shell, that is a electron cloud. In the electron first shell, that can occupy 2 electrons. The second shell can occupy 8 electrons. So it's a octet, it's a stable. Third also 8 electrons. So the valence electron is the outermost electron. This one right now I draw is a stable. If it's valence electron, like we mentioned in the oxygen or hydrogen, oxygen need 2 more. Hydrogen, 1 electron. So the valence electron determine the bonding behavior. So you need one. This oxygen need 2 more. This hydrogen need 1 more. So this valence electron, that is a valence electron outermost shell. Then we look at the chemical reactions, type of chemical reactions. So this is anything, chemical reaction is the one in your system, your human body. We always having that chemical reactions. Our body accumulate with the chemicals, protein, DNA, enzymes, making some proteins, breaking down things. Whatever we eat this morning, carbohydrate is breaking or building certain things. Always going through that chemical reactions. They are chemical bond made synthetic, which is called anabolism. The making something, making new thing, making new protein, that is synthetic or anabolic reaction. Broken, which is a decomposition or catabolism. That means breaking down something. So when you eat a carbohydrate this morning, it has to break it down to make energy. So synthetic or anabolism, the reaction thing is a reactant, A is a reactant, B is a reactant, this is a product, this is a chemical reaction, it's a one-way reaction. So here decomposition, this A, B breaking down into what? A and B, which is a product. Redox reaction, transfer electrons or atoms. The chemical reactions can be either that they always redox reaction. Reduction and oxidation reaction. One is going to reduce, one is going to be oxidized. One is going to gain the electron, one is lose the electron. So chemical reactions, they can be atom, ion, molecule or compound, interact to form or break chemical bonds. So in this case, form a bond. So here it should be bond, it should be A to B. So here the bond has to break it down into A and B. Reactant, this in this case A and B are reactants. This is a product. This one, in this reaction, AB is a reactant, this one the product. Then make sure you know this one, metabolism. This is a collective term used for sum of all the anabolic and catabolic reactions in the body. That means all the reactions happening in the body, if you put the term or the name, which is called what? Metabolism. Some of you guys metabolic rate is high, some of the metabolic rate is low. Depends on the age, their physical condition and things like that. Synthetic reaction, which is we talk about anabolism. So anabolism is what? Making something. You can see this is a reactant A, this is a reactant B, then you making the product C. So when you go through that making something, they go through the dehydration reaction. Dehydration means what? Losing water from that reactants. So here this hydrogen and oxygen combine, they is a byproduct. Water is a byproduct of this reaction. Then what happened? This bond between the carbon and nitrogen is forming over here. There's a bond is forming. So they're making that a product. So this is a byproduct, which is synthetic reaction. Then when you look at decomposition reaction or catabolism, this is a disaccharide. Di means two carbohydrates. Then water is there. You need water to what? Making that a hydrolysis reaction. So this one, they adding the water to that reactant. So when you're adding the water, this become that a product is a breaking down. This is a glucose and glucose, which is a dehydration reaction. Decomposition, in order to decompose or catabolic reaction, it go through that hydrolysis reaction. Then next one, we're going to go over enzymes. What is an enzyme? This graph you see when we study about enzyme. So enzymes are proteins, most of them. So these enzymes are what? Helps for that reaction. We studying about reactions, how they helping. See, in order to make A plus B to C, take some time, maybe 1 hour. Maybe think about you making a cake. You put all that ingredients like sugar, what else? Oil, flour, egg, all this mixing. These all things are reactants. Then you put it in an oven for a certain time, maybe 1 hour. Then you make what? Cake. Cake is what? Product. For example, here A plus B to C, making the product, you need 1 hour. Think about it. Then you need to put energy. You put in an oven, you using electricity or gas. That's energy using. So energy required for. More time is take what? More energy you adding to that reaction. For that reason, so this enzyme helps to what? Bring it down that activation energy. How much energy you use it to make this product going to be low if you are adding that enzyme. Think about when you make a cake, you take 1 hour, is something there to make that 30 minutes. That would be great, isn't it? You're not going to lose a lot of gas, energy, and time going to be, you're not wasting that much time and energy. So here these enzymes, what they do, they minimize that activation energy. In order to make that reaction, the activation energy to make the reaction, reactant to product is going to be low. For example, here, this graph is very easy to understand. This is a progress of reaction. So reactant and product, no difference. You see that same reactant, same product forming. So energy is going to be that in y-axis is showing that free energy. So reactant and they started the same time for these two graphs. So one is go here and come back and making that product. One is going here in making the product. So this one taking what? More energy, using more energy. So the time also increased because of that the case. So energy this much. So what happened? This is a progress of reaction without enzyme. Without enzyme is what? Taking more energy. So with the enzyme it's taking less energy. So that's why it's called what? This minimize that activation energy with that enzyme if you are adding that enzyme. So catalyst, a substance that increase the rate of chemical reactions without being permanently changed or depleted. So it's not going to change anything. Even using the enzyme, it doesn't use that to change anything. The enzyme is going to be stay like that. It's not going to change the enzyme character or anything. So we can read that one, protein catalyst that increase the rate of chemical reaction by lowering the activation energy necessary for reaction to begin. So other influences on chemical reactions, temperature. Think about if you're adding salt into the water, you mix it. So in order to dissolve all the salt into the water, better do that. The water is if you put in that stove, temperature increases, that easily can dissolve. So temperature can what? Increase that chemical reactions faster, quicker. Then concentration of reaction. If you are adding more salt into the water, then it's going to be what? That increase the chemical reaction. Next thing we looking at the water. What is the water important for the water? We have to know the properties of water. They have certain properties. They are universal solvent. Our body is most of the body is what made with the water. So it is there are some characteristics for this water, some properties along with that. It does have polar nature. Polar means it's as a charged particle, like I mentioned that earlier, H2O. So partial negative, partial positive charge, so it's a little polar. So it has the ability to interact with other things. They are hydrophilic. So philic means love towards something, love towards something. So the philic, what is that? Love towards, in this case, water. Substances attracted to water, water loving, can dissolve in water. Hydrophobic substance does not attract to water, water fearing. Phobia means what? Fear. So for example, think about adding water into adding oil into the water. Take a beaker, water, then adding some oil. What happened? Oil is float on water, it never mix it. So for example, that is Caesar salad dressing. You ever seen that salad dressing has a oil on the top? So what is it? It doesn't mix it. You have to shake it in order to mix it. So this is what? Hydrophobic. Oil is what? Hydrophobic. You cannot mix it. For example, salt is hydrophilic. When you're adding the salt into water, you can mix it. Then other things, cohesion and adhesion properties. They have attracted to that water to water molecule. That's why it's going to stuck in that water molecule. You can take it in a bowl and other things. Then also, how do you drink? How you use that straw to drink some water? Because the water also, think about this, a straw. Water is also attached to that other object. In this case, a straw. So that's why you can drink it. You can use that straw to what? Taking the water away from that wherever they have it. So then other one, can cohesion, attract one water molecule to another water molecule. So that means H2O2, H2O, that is attraction to other molecule. That means this is the one, any object. So 92% of blood is composed of water. Blood has what? Other things like red blood cells, white blood cells, and etc. Then properties. they have stabilizing the body temperature. That's very important because when your body is heating up, when you run or fever, what happens? You sweat. So cool down the body. So they when evaporate water, sweat evaporate is cooling down. Protection. They act as what? Lubricant. Reduce the friction in between the joints. So cushioning liquid, that is synovial fluid, that is very important. Then inside the serous membrane there are some liquid inside. That's all protecting, cushioning internal organs. And participate in chemical reactions. We see that water is very important for that hydrolysis reaction. They are adding that water to that chemical reaction, so it can form that product. And other things you can read it: universal solvent, temperature regulation, hydrogen, forming the hydrogen bond, polar molecule, all these things we mentioned. These all are properties of that water. That's the reason water is very important to study and know what is their characteristics and properties. Then we looking at that acid, base and buffers. What are those things? So you heard about pH scale. pH scale is start from that 0 to 40. We come closer to the 0, what is that? More acidic. We're going closer to the 14, that more basic. 7 is a neutral. That neutral, when you look at that here, water, distilled water, DI water, not the faucet water, distilled water has a neutral 7. Then your blood pH is going to be what? 7.4, little bit more that acidic. Then acidosis is what? If you less than that 7.35 is acidosis. Alkalosis is that greater than that 7.45. Organic versus inorganic compound. Organic is what? Anything contain carbon. For example, big molecule like carbohydrate, proteins, lipid, nucleic acid. That doesn't mean carbon dioxide. Carbon dioxide is not organic, even though it does have a carbon. So then inorganic, no carbon, water, salt, this. We're going to go over that carbohydrate. Carbohydrate is a sugar molecule. They can be monosaccharides. When you see this root name saccharides, they are related to carbohydrate. Mono means one, di, this again that root word. There is another root word over here, same thing. So all carbohydrate. When you see that, you know they're talking about sugar and carbohydrate. Di means what? Two. Poly means many. So here example of monosaccharides: glucose, fructose. Disaccharides, that means two of them. See, disaccharides are what? Two of them. Two of that monosaccharides put together make a disaccharide. This is a monosaccharide. This is a one. Glucose is monosaccharide. Fructose is a monosaccharide. They put together is making that disaccharide, which is a sucrose. Polysaccharides example starch you heard about. Rice has a starch, potatoes has a starch, cellulose is in plants, glycogen is in that stored form of carbohydrate in animal. So this is a poly, many, see you can see each chain, this is like this, they are monosaccharides. The monosaccharides put together making that polysaccharides. You can see different shapes. Cellulose is in fiber, this is in the plants. Lipids containing carbon, hydrogen, oxygen. Also you can see sometimes phosphorus. Lipids are what? Hydrophobic. Like I said, phobic means what? Fear of water. Because of that, insoluble in water. Lipids, what they do? They use as insulation. So the fat is around your internal organs. The cushioning. Then also energy storage. Triglyceride is example of lipid, which is energy storage. Phospholipid, that is example of lipid, which is see that cell membrane. We're going to study cell later chapter. This is a cell, this is a cell membrane. They're made with the phospholipid. Then steroids, which is hormone, cholesterol. They are steroids, are examples of the lipid. Then prostaglandins, echinocinoids. These are all examples of the lipid. They can be saturated or unsaturated. Saturated is a single bond. All this, like I mentioned, this lipid, carbohydrate and everything organic, they made with the carbon. So think about it, this is a carbon chain. All this carbon single chain saturated with the hydrogen. Unsaturated fat, they have a double bond. For example, carbon. See here, everything is saturated, but they should have a double bond. So here, so this one has to be removed. I can have one more. Hold on. Hydrogen. So 1, 2, 3, 4. Then carbon. Then think about another double bond. So it does have already 4 bonds. So it doesn't need anything. Maybe this one has one more hydrogen and hydrogen. So it's a double bond is formed. That is unsaturated. So we're going to see that lipids examples. Phospholipids is very important for that cell membrane. When you look at the cell nucleus, is a cell membrane. The cell membrane has a phospholipid. They have a hydrophilic region and a hydrophobic region. This is a hydrophobic tail. This is a hydrophilic region. It's a polar region. So then other one has that glycerol. Fatty acid has a glycerol backbone. We can see over here fatty acids. Then triglyceride is forming. Saturated, I show that one. So every carbon was saturated with hydrogen. There are some double bond here, which is unsaturated fat. Proteins are very important for our system. Those hormones are proteins, enzymes are proteins. They made with amino acid. They are basic building block of proteins. Amino acids are what? Building block of proteins. Then forming the bond between the amino acids are peptide bond. For example, think each pearl is a different shape and things. Each one is amino acid. Maybe this one. This is a different amino acid. There are 20 amino acids, each one forming bond between each one. This amino acid to amino acid. So this one, this is a protein. So in order to make a protein, this amino acids have to form a bond. This bond we call the peptide bonds. So you can see that anything making that dehydration reaction, this is losing the water to synthesize something. They're making that anabolic reaction, synthesizing that peptide bond and making that a protein. There are 20 amino acids. They starting from the primary, like a primary, then is going to that quaternary. That is example hemoglobin. Then another thing about the protein when you study, it's a denaturation reaction. Denaturation is the one loss of structure and function due to pH or heat. So for example, egg. You're making a egg sandwich or egg, maybe sunny side egg. You put that on a pan. When the temperature is going up, the transparent liquid part of that egg become white color in structure, so that you eat. So the white is what? Protein. So the protein that when you making that you putting in that pan is lose their function and the structure. You cannot bring it back that original stage. That happen due to what? Heat. So that is a denaturation, is a loss of structure and function. That is very important. If your body temperature is increasing, maybe you have fever, what happened? That your system has a lot of proteins in order to make a reactions and things go on. But if your temperature increase, it can what? Mess up your system. You can change that configuration or the structure of that protein. That process we call denaturation. So this is a primary structure of the protein. It's like a pearl on a string. Each one can be different amino acids. Then it can be a secondary structure. Pleated sheet or helix coiled around like this. Then third one is tertiary structure, folding interaction with the polypeptide chains. They have some interaction between that, certain regions. Then quaternary is the fourth stage. So these are different individual subunits of this structure. This is a one protein, this is another one, this is another one, this is another one. But they interact each other and forming this kind of stuff. So this is the example of quaternary is a hemoglobin, which you see in that red blood cells which carry oxygen. Then we're going to talk about the nucleic acid. Nucleic acid, DNA, RNA, and adenosine triphosphate, which is ATP. DNA is a genetic blueprint of that any organism, plants, animals, fungi. RNA is a protein synthesis. RNA is very important for that protein synthesis. DNA have a blueprint, then it's going to make that DNA. So the DNA is a double-stranded structure. It converted into single-stranded or RNA. Sometimes can be double-stranded RNA, DNA, RNA, then it's going to protein. This is a central dogma of biology. This process going from DNA to RNA is called transcription. Then going from here to here is a translation. So ATP is energy currency for our system. So this nucleic acid that as a nucleotides are made by sugar, phosphate and base. See this deoxyribose, they are sugars, deoxyribose sugar and ribose sugar. Deoxyribose sugar you see in DNA, RNA you see ribose sugar. So this is the deoxyribonucleotide. Nucleotide has a deoxyribose sugar, nitrogenous base, see sugar, nitrogenous base and phosphate group. Then give that nucleotides. So this is deoxyribonucleotides because it's have deoxyribose sugar. Otherwise, just a nucleotide, it should have just what? Ribose sugar. So these are the nitrogenous bases, guanine, adenine, guanine, A, G, guanine, then cytosine, thymine, and uracil. So these all are here. This is a pyrimidines, this is a purines. Purine has a double ring structure. So one ring is another ring. So this is a single ring structure. Nucleotides is composed of penta sugar, 5 carbon sugar, either deoxyribose or ribose sugar. You see that in DNA, RNA. Then nitrogenous bases, either one of these nitrogenous bases and a phosphate group. So genetic material of cell copied from one generation to next. So DNA is composed of double-stranded structure, 2 strands of DNA nucleotides. A and T goes together, G and C. They held together by hydrogen bond. They have a double helix structure form. They are anti-parallel. One goes one direction, another strand goes to the different direction. So they forming that sugar phosphate backbone. This strand, then yet another definition gene. So sequence of bases that code for synthesis of RNA or protein. So genes in the DNA structure, one maybe region is for code for certain gene, maybe color of your eye, maybe your how much enzymes you produce, like insulin. So it's code for certain thing. This is a genes. This is if you need more insulin, you ate a lot of carbohydrate, you need more insulin. Only gene expression going to happen this particular region. Not all of them going to express. Only this one expressed when you need insulin. So it will express and making that insulin, the protein. So that's why I say it's going from that insulin, which is a protein. Now to make this DNA, gene expression happen, going to that RNA, then it will go into that protein formation. So the structure of the DNA, double helix. So this is a backbone we talk about, phosphate and sugar. This is a nitrogenous bases, one of these cytosine, thymine, guanine or adenine, which is DNA. DNA has all four of them. Uracil we studied earlier, but uracil you see only in that RNA. So here that nitrogenous bases, adenosine goes to what? Thymine. So we're going to see that here this one, we mentioned this is anti-parallel. That means these strands, this is this strand, this is this strand, taking portion of this. This is what you see over here enlarging. So here this is a backbone made with the phosphate and sugar. So this is the way arranging. This is a 5' end. This is a 3' end. It should be reverse over here, 3' to 5' end. It's a, that's why I say it's a anti-parallel. One goes this direction, one goes this direction. So in between, you see nitrogenous base connected by that hydrogen bond. This thymine goes with the adenine, always A and T. So C and G. So it's very important. The space between this has to maintain. One double ringed structure has to be what? Related to that, connected to that single ring structure. So that's why it's maintaining that distance between two strands. Think about if you have two double stranded here, the distance between this is going to be varies. So adenosine triphosphate, which is adenosine here, then the adenosine is sugar and nitrogenous base, then phosphate group. So in that ATP, triphosphate should be 3 phosphate group. So 1, 2, 3. So here this is adenosine triphosphate. If we lose one of the electron, it's become what? Adenosine diphosphate. This is what? Provide energy for all chemical reactions. So when energy, when ATP is not available, then there is no energy. You don't have any energy, you stop all the chemical reactions. So adenosine triphosphate. So catabolism of glucose. Glucose is a 6-carbon molecule, C6H12O6, 6-carbon molecule, breaking down and making the ATP. That means it's a breaking down all these carbon. Then breaking down, they release what? Energy. Then that when they breaking down, release the energy to make what? ATP. Then then this ATP has full of energy. When each phosphate group is a release, a breaking down is release energy into that system. So ATP breaking down to ADP is a phosphate, one phosphate group you get, then also release energy. Now to make ATP, you need ADP, adding phosphate group and put some energy into that system. So this is what we study this chapter 2. If you have any questions, please let me know. Okay.